We finished module 1 of the course, which is on gene expression. So I would like to give a summary of this whole module and the topics we covered. We started explaining high throughput sequencing. Currently, the technology leader is Illumina, and so we only teach you how to analyze data from the Illumina sequencing machines. And we learned the FASTQ format, which is the raw data that's coming from Illumina sequencing. And the very first step you might want to do is quality control. And we discussed FastQC. This is irrespective of whether you are sequencing DNA or RNA. You can always use FastQC to generally look at the sequencing quality. Then we discussed alignment, right? There are different alignment algorithms. And we focused on BWA, which used the Boros-Wheeler transformation and LF mapping in order to identify each read where it comes from in the genome. There is a competing algorithm called Bowtie, but we only have time to discuss BWA. They have very similar ideas behind. Both use BWT. And with BWA alignment, we will be able to find millions of reads very quickly where they map to the genome. And the format you get is a SAM format, which provides the read quality, the location of the mapping, and whether there are mismatches or small gaps in the alignment. And then this can be converted into a BAM file, which is a binary format of the SAM, and it's a lot smaller. And then there is BET format, which is even simpler format of the alignment, although there is information loss. Whereas with BAM file, you have all the information as FASTQ, as well as the alignment information. And we briefly explain SAM tools, which can help you evaluate the SAM data to look at read mapping quality and also the percentage of mappable reads and so on. Then we very quickly move to RNA-seq and gene expression. Again, the experiment goes to the RNA-seq experiment after we get the data is in FASTQ data. But here, in order to do the alignment, we don't really use BWA for mapping because we need to map it to the RNA instead of the DNA, which has splice junctions. And so we use splice-aware algorithms. And HiSat is the algorithm that was based on Bowtie, which is using the Boros-Wheeler transformation. But currently, STAR is considered the most accurate one, also the best balance between memory usage and speed and accuracy. And this one actually uses a suffix array. We focus the course on the alignment to the reference genome, but there are also de novo RNA assembly algorithms, which does not even need the reference genome to be available. Supposedly, if you have a new alien, like if you found some alien, as long as we know this alien has DNA, you could sequence its RNA and potentially use Trinity and cufflinks to assemble the RNA transcript and get abundance at the same time. So for the course, we focus on the very basic things. So we just use STAR for alignment, and then we need to do quantification. For quantification, the current best, most accurate approach is RSAM. So we talk about different way of representing the quality, or sorry, the quantification, whether you use normalized read count, which is usually CPM, which is count per million. So this only normalized by library size, or the FPKM or RPKM, which also normalized by the gene length, or you can normalize by TPM. And the difference between FPKM and TPM is whether you normalize by the library size first or whether you normalize by the gene length first. And apparently this makes quite a big difference. And currently TPM is considered the gold standard in terms of, or the most accurate in reflecting gene expression levels. So RSAM is very good, but it takes a lot of time. Therefore, there are pseudo-aligners such as Salmon and Callisto. These two also have very, very similar approaches, which only align the reads to the known annotated transcript, which only accounts for 1 to 2% or maybe 2% of the genome, therefore can be done very, very quickly. And so you will get the expression, a readout for both normalized read count, FPKM and TPM from both Callisto and Salmon. And these two also give very, very similar results. And it's much, much faster. Besides, these algorithms can run either directly on the FASTQ file, or if you already have the alignment, you can also run Salmon and Callisto on the BAM files from a star alignment. After we get the quantification, we will want to compare the differential expression. And we discussed this, and in RNA-seq, the read alignment distribution across the genome actually follows a negative binomial distribution. And there are different also differential RNA-seq analysis algorithms. If you have limited number of samples, DE-seq is currently considered the most accurate one. It calculates differential expression at the normalized read count level. And it also uses hierarchical modeling to shrink the variance by borrowing information from nearby genes or genes with similar expression levels, so that if you have limited replicates, in order to estimate a variance accurately, it can kind of look at other genes with similar expression to get a more accurate expression. In addition, both DE-seq and EdgeR or Zoom, Lima-Voom, they use ways to reduce the total number of genes that you need to evaluate for differential expression to reduce the multiple hypothesis testing. We didn't cover in this course Lima, which also is a widely used algorithm. And it calculates differential expression based on the count per million. So it's just kind of also a normalized count for differential expression. Yeah, and so both DE-seq and Lima can shrink or stabilize the variance using hierarchical modeling. If you only have limited samples, DE-seq, I would say, is probably the choice to go. But if you have, for example, very complicated experimental designs with many different conditions that you have to compare with, or if you have many, many samples, you can use Lima. And even if you have hundreds of samples for two conditions, these algorithms will actually give you very, very similar results. For differential expression, we always face the question of how many genes we want to report as differentially expressed. Since there are tens of thousands of genes that we have to evaluate, we run into the multiple hypothesis testing issue, because every statistical test done on a gene has a small probability of making a wrong prediction or wrong call. If we're evaluating tens of thousands of genes, the chance of us calling something incorrectly would be quite high. And so we want to determine the number of differential genes to report to the user by correcting or by limiting the total number of mistakes that we want to make, or the percentage of mistakes that we can afford. So Bonferroni is a family-wise error rate that you don't want to make even one mistake or control the likelihood of even a single mistake. And it's a very, very conservative correction approach. Most of the time with RNA-seq or actually any genomic experiment, people use false discovery rate. And we discussed the Benjamini-Hochberg approach for correcting the multiple hypothesis by looking at the kind of distribution of all the tests that you do and use those to estimate kind of the baseline, because we know by chance the distribution of p-values should be a uniform distribution. And therefore, if we assume this is all the other tests will give you a baseline uniform distribution. And then if there are more genes that give you a smaller p-value, more than expected by a uniform distribution, we will know roughly what is the percentage of the calls made by chance versus how many are likely to be real. And so that gives us a false discovery rate estimate. So most of the time, if after FDR adjustment, you only have, say, 200 genes, you should probably just take all of them. But if after the FDR adjustment, you still have, say, 1,500 genes, you could use fold change on top of the FDR to cut the differential genes. Say we want all the genes that meets FDR 1% and a twofold change to get you to the more robust differential expression. And because for a lot of the later analysis, you probably want to control your total number of genes to a reasonable number, usually less than 1,000 genes. Especially for gene ontology or pathway type of analysis. Next, we discuss clustering. Because there are hundreds of genes that we're reporting as differential, to a biologist, it's very difficult to look at the gene names and understand what's going on. Therefore, we first try to cluster the genes based on their differential expression, differentially expressed genes. And especially this is useful if you have multiple conditions. Let's say a time course drug treatment, you use the clustering of all the genes to look at the overall behavior of genes over the time course. And so there are different clustering approaches. We can use hierarchical clustering, which kind of always start merging the things that are closest to each other, and they keep on, to create internal nodes and then try to merge the remaining ones that are closest to each other. We can also use k-means, which is kind of a disjoint clustering, and every gene either belong to one cluster or the other cluster, whereas hierarchical clustering, eventually all the genes will belong to the root node. For k-means, we discussed, potentially determining the K, but the reality is usually most people can only deal with K smaller than 7, because if it's more than 7, a lot of people just focus on the 7 that they can absorb. However, if you only have very simple conditions, say if you only have 2 conditions, with drug treatment, without drug treatment, you probably don't even need the k-means of 7, because you just ask, what are the genes upregulated? What are the genes downregulated? K-means of 7 would be useful if you have, say, a time course of 5 times. And so the 2 to the 7 would be a lot, right? Then you want to use k-means. Another way to do clustering is to run PCA. And by this, actually, you could run PCA on the genes, but you could also run PCA on the samples. In this course, you probably see more cases where people use each sample to cluster the samples using PCA. And so PCA, in addition to being a clustering approach, it is also a dimension reduction approach, which try to use the top principal components to capture as much of the variations of the data as possible. And always the earlier principal components capture more variance than the later components. But yeah, so these are the different clustering approaches. Once we run clustering, you might find that the data have batch effect. And this kind of go back to the analysis. You might want to do a clustering right after the quantification. So before you do differential expression analysis, because without batch effect removal, if the samples are not clustered well, your differential expressed genes may not be accurate either. And so using the clustering of samples, you might be able to detect the batch effect. And you can see that either from hierarchical clustering or PCA that, oh, there's batch effect that you need to remove. And there are different batch effect removal approaches. And we mentioned briefly intuitively, you can imagine batch as an additional treatment. And you can kind of look at what genes are differentially expressed between this particular batch and kind of remove that out, like eliminate this as kind of a principal component in some sense, and then look at the remaining gene expression. And so both Lima and Combat can allow you to do the batch effect removal. Currently, if you have a simpler batch, Combat is actually really, really good for batch effect removal. But if you have a very, very complex batch, for example, we have one study where we have both fresh frozen and formalin fixed FFPE samples. And also the samples are processed in different days and also in different hospitals. It's very complex, which means actually you have different ways of combination of different batches. Lima is probably better, and you can run the log TPM to remove the batch effect. And it will actually generate a matrix for you, which has the batch effect removed data. Then you convert the log TPM back to the original TPM for analysis. And so usually I would say for that, you would probably use Lima, sorry, CPM is okay, TPM is, yeah, these two are okay. But for DE-seq, you can also run DE-seq with simple batches, but those will only give you the differential genes, and it will just consider this particular batch as a variable that it needs to normalize out. And you will not really get a normalized matrix for downstream analysis. So if you also need to, say, draw PCA plot or do other classification, you can still run DE-seq to get the differential genes, but you will not get that matrix to do the downstream analysis. So for RNA-seq, sometimes you might also want to do classification. So in this case, if you have a lot of samples that you have already profiled, some are, say, normal, some have disease, and then you have a new sample that you have RNA-seq data, you want to see, does it belong to the normal or the disease? Or in some cases, you might have a subtype, right? There's a lung cancer has adenocarcinoma and squamous lung carcinoma. They both belong to non-small cell lung cancer, but there are still slight differences which might influence their treatment, say. And so in this case, you want to use classification, right? Some samples are labeled and you want to figure out the result for the new unlabeled sample. And so that is the machine learning. Sorry. By the way, after you finish clustering, you want to do some annotation, right? So this is based on, say, differential gene cluster. After you get the k-means cluster, for example, you can run gene ontology, for example, David, to get the gene ontology annotation. You want to basically, instead of giving the biologists hundreds of genes, you can say, okay, this is related to the PI3 kinase pathway or something like that. Or all the genes are related to a particular, say, mitochondria genes located in a specific cell location. And then we also talk about GSEA. This is useful in two different ways. One is that if you don't have a sufficient number of differentially expressed genes, because many genes might only be slightly differentially expressed. So collectively, they might have a trend, but at individual gene level, maybe none of the genes show you a strong enough statistical significance as being called to be different. And so GSEA is really useful there. In addition, over the years, the group that developed GSEA have looked at many differential gene expression data to generate gene signatures. And so if you identified a group of differential genes of some conditions, you run the GSEA, and it can actually use the other signatures to kind of tell you, you know, somebody else have done a similar expression and they saw those genes as differentially expressed, which is similar to yours. And that kind of gave us some idea what my experiment is really different, or say if this is a drug treatment, and I do the differential expression, and based on the previous GSEA studies in the signature, I might be able to learn what other experiment also see the same group of genes perturbed and understand my drug perturbation better. So for machine learning, yeah, we mentioned there are unsupervised machine learning approach and supervised machine learning approach. Unsupervised is we don't look at the sample labels ahead of time. You just try to cluster the samples. And this clustering, you can use just hierarchical clustering. You can use MDS. You can use PCA. So MDS and PCA, remember, these are also dimension reduction approaches. And so after the cluster, then you say you label the one class with one color and you label the other class with a different color. And then you have your unknown sample and you just look at whether the unknown sample is closer to the, you know, which cluster it belongs to. Then you try to make that prediction. It has been used, but this really rely on the data being perfect, you know, like everything already clustered very clearly. Sometimes, depending on the noise of the data, is not so clearly separated. And so we might want to use supervised machine learning approaches, which we try to identify features that can really separate the samples between the two classes. And so there are feature selections involved. And there are also a number of different algorithms that are under development. The first is ICA. This also is a dimension reduction approach. And remember, we try to maximize the center, the two class center distance and reduce the within group variance. It's not as much used in biology, actually. And also there is logistic regression, which is used quite a bit. And we run into the problem of, so logistic regression, basically you give genes different weight and you're trying to see, you know, like in this logit way, whether the sample belong to one class or the other. And the nice thing is that it gives you a probabilistic assignment. So some genes, or sorry, some samples you're very confident, other ones, you know, you are close to the 50-50 boundary. Also because logistic regression give gene a different weight, and so you can see what genes are really being selected to really separate the two classes. With logistic regression, because we are evaluating so many genes and there is the chance of overfitting your data. So regularization is a way to not give too high weights to the genes in terms of you know, like you're assuming that these two groups have no difference, and you try to favor that and really call as few genes as possible as really differential between the two groups. And the ridge regression, lasso regression are just different ways of doing regularization. Ridge regression is doing the L2 penalty. So the penalty is assessed as the squares of the coefficient, whereas a lasso is L1 penalty. So the penalty is assessed as just the slope level. And the elastic net is kind of a mix of the two, and you evaluate how much you use ridge or lasso based on your cross validation. And then k-nearest neighbor is kind of a black box, but you know, sometimes it works very well. You just guilt by association. For every sample, you just look at other samples that look similar, and then you do a voting to decide whether this new sample should belong to the class 1 or which of the group it belongs to. It's a very simple approach, but it doesn't really provide a lot of insights. And then random forest, I would say before the convolutional neural network or these deep learning approaches, random forest is probably the method that works the best. And it basically is a tree-based approach. Every time you find a kind of a node to divide the samples into two groups, and then you find a cutoff to separate the samples so that you can reduce the Gini impurity, basically to have better separation of the two group of samples. And so every stage you try to reduce the mixing of the samples. Nobody actually used decision tree. However, random forest is basically you sample the number of genes and you sample the number of samples to run decision tree many times. And then at the end, you see which feature or which gene is being selected for most of these trees. And then you use all of the forest, everybody to make a decision. It's kind of a wisdom of the crowd. And then actually works fairly well, and it also give you some hints of which feature is important. And then finally, there's support vector machine. I frankly don't see it used as much in I mean, you can try it out, but I don't think it's used as much in biology. But these are all just different ways of separating the samples and between the two classes and try to gain some insights. With support vector machines, like there are different kernels you can project your data to, and I think the decision is also you try different ones and use cross-validation to decide which one will give you the best one. And you hope that this will give you the best one. But I think in terms of intuition, it's probably not as good as logistic regression or random forest. So that kind of covers the main topics that is in the first module. From previous years of teaching, we realized that this course, because we cover a lot of material, lecture by lecture, sometimes the students throughout the course understand all the individual steps. However, they don't know how to string the things together in order to actually deal with a real data set. And so we want to show you in the review lecture how to really integrate the things you learned in this course together when you are dealing with your own problem, so that besides doing the homework in the future, when you have to use bioinformatics on a real data, what's the general strategy? So the general strategy is the first step. You do the pre-processing on quality control. In high throughput sequencing, this usually involves, you do some read alignment, you just get some numbers, and you make sure the batch effect is removed. You make sure your RCQC, quality control the RNA sample is okay, and the basic things are done. You just at least can get some data that you can use. The second step is really more, I would say, so by the way, the first step potentially is also more computer science related. Well, I guess batch effect is more CS, is more statistics, but alignment, just doing counting, these are usually just CS. But the comparison, you're dealing with more quantitative issues, right? Batch effect is also quantitative. You're dealing with comparing numbers, and that's when statistics become more important. And you have to model the data distribution. Does it follow normal or negative binomial or chi-square and so on? And also correct for multiple hypotheses. You try to learn information from the adjacent genes or nearby genes with similar expression. And so those kind of are more statistical. And so the comparison can already give you a lot of interesting biological insights. And after that, you might want to do some visualization, draw some plots, generate some tables, annotate with gene ontology or pathways. But this is still mostly focused on just looking at your own data. Later on in the semester, we didn't actually show you this downstream analysis part as much yet. But later on, I hope you will see that there are actually a lot of public data in the public domain, and being able to use the public data in order to do annotation, in order to help you with understanding is extremely informative. It can also help you prune your hypothesis. And also you can do more things like classification or machine learning, which is kind of a fancier things, downstream analysis. So usually for any informatics question you have, you might want to separate them into these three categories. The first is, just pre-processing, QC, get some basic readout. Second is usually two groups or three groups, or comparisons, looking at differential things, do some basic annotation. And the third is try to use more public data and more sophisticated methods to understand mechanistically what's going on. So I want to use a few scenarios to help you understand how you actually analyze some data. Let's look at this first scenario. We observed some drug effect on cancer cell growth. So these cells are growing in Petri dish. You treat the cell at, say, different dose or different time points. You can see, eventually it's not, the cancer cell doesn't like the drug and eventually will die. And so you say we generated RNA-seq experiment on the cancer cell with or without the drug. And also, without the drug is just, 0 day. And 0 hours. And with the drug, you have 1 hour, 5 hour, and 24 hours. And let's just say you generated 3 replicates in each case. And the 3 replicates are 3 biological replicates done on 3 different days. And so let's think you have, at the end, many FASTQ files. How do you really deal with this data? So think, like we want to do the QC first, right? You do read mapping, and you might want to do some RCQC to make sure the RNA samples are okay. If there are some really bad outliers, you might want to remove that. If it's cell line, I would say most of the time it's probably okay. And then you try to get the expression index, right? Using, say, Salmon or RSAM. And then you probably want to cluster the samples because remember, these are 3 biological replicates done on 3 different days. And so we want to see whether the samples are clustered by, ideally you want to see the samples are actually clustered by the with or without drug treatment, and also with the time. Ideally the 3 no treatment are together, 1 days are together, 5 days, and 24 days are each time point are together. But if that's not what you see, and you clearly see the 3 days as being the major variable, you might want to remove that batch effect. And then you want to try the clustering again to, double check your batch effect again. After you remove that, you can look at the differential expression, 1 hour compared to 0, 5 hour compared to 0, 24 hour compared to 0, to look at what genes are differentially expressed. And then you can run DESeq2, or you can also run Lima to get the differential expression. And then based on that, you might want to run the clustering. And since this is like 4 time points, you might want to run a k-means clustering to see whether there's like early time up genes later, like for example, some genes might continue to go up, some gene might go up initially and come down, and some gene will continue to go down, and some gene go down and maybe come back to normal, and like so on. And so you can group the genes by their behavior, and you can visualize this on a heat map. And then you can annotate each of the clusters separately on a gene ontology, and then see the differences. You can also use public data based on the differential genes just from 1 hour differential gene or 5 hour differential gene, 24-hour differential gene. You can run a gene set enrichment analysis. This is in gene set enrichment is a little bit of using public data to see what else have what other experiment have seen the similar genes to be differentially expressed, and what do people know from those studies about those group of genes. And so that gives you some insight from the public data. And hopefully this will give you some idea what this drug is really doing at the mechanistic level. Okay, so in this course, because this year we're not going to have a final exam, but in previous years, we had final exams and the final questions were like this. When you are given data like this, we give you a table and we ask you, roughly how you would run this. And you might want to just say, okay, first I use STAR, I do the mapping to get the map reads to the genome. Then I use RCQC to QC the RNA-seq results. You can use Salmon or Callisto to get expression index. You can also use RSAM, of course. And also you want to use PCA or hierarchical clustering to visualize the potential batch effect of the samples and also get rid of outliers. Then you want to run DE-seq or Lima to get the differentially expressed genes at later time points over the 0 hour. Removing batch effect if needed, because if you only run DE-seq 2, you will just get the differential genes directly and you won't really see the matrix again. But by the way, so for the PCA, you might want to run it before and after the batch effect removal to make sure you have removed it, right? And then you might want to do some k-means clustering on the genes and not on the samples, just to look at how things cluster. And also you can use heat map to visualize the differential genes. And then you can run David or some other, there are like gazillion gene ontology tools. We just thought David is one. And there are also GoStat and other things that you can run directly from Bioconductor and find the genes or pathways that are enriched in different clusters. And then use GSEA to find other public data when the same group of genes are observed in disease or other perturbations. And so, yeah, we'll do more on public data reuse later in the semester. So I hope this gives you an idea when you have a data like this, how do you use all the tools that you have learned in the first module to, at least get to a reasonable understanding. Let's look at another scenario. Supposedly, we have RNA-seq data of 200 pairs of lung cancer samples. These are all diagnosed as adenocarcinoma, non-small cell lung cancer. And we also have their adjacent to normal tissues. And let's just think about this really ideal scenario. 100 of these patients, 5 years later, after surgery, within the 5 years, you follow up with the patient and you saw 100 of these patients relapse, whereas the other 100 patients, they didn't relapse. And we're like, oh, that's great. You learn something and you want to see what's the difference. And so now you have 5 new RNA-seq samples. And we know that they're also lung adenocarcinoma patients, and we want to decide whether they will relapse five years later, because if you predict that they will not relapse, maybe the surgery would be good enough. Whereas if you predict, oh, these patients are going to relapse in 5 years, then maybe after surgery, they might also consider treating the patient with chemotherapy to reduce the chance of relapse. So again, this is like 400 FASTQ files. So a lot of data to deal with. So what do we do first? Again, we will do read mapping. FASTQ, sorry, not FASTQ, DE-seq2. Frankly, when you have a lot of samples, sometimes you might want to just run a Salmon And then do a clustering. And if the data is good enough, like just based on initial analysis, you see that, because a lot of times the bad quality samples in the PCA will show as outliers. You could potentially just run salmon and then run PCA, and you say, okay, these samples look pretty good. Clearly there's a separation between cancer and normal, and we can just forget about, and then you can just run RCQC and RCQC just on the few outliers, and you decide, oh, these are clearly outliers. You can just remove them as bad quality samples. So that will save you some time. RCQC unfortunately doesn't directly run on FastQ. It runs on the BAM. That's why we still need to do a little bit of a BAM, like a star mapping. One way to do that faster is you don't need all the reads to run star. You can just subsample, say, 5 million reads. So supposedly each sample is sequenced 50 million reads. You can just get 5 million reads to run star and to run RCQC, because it will give you a pretty good idea what the quality of the sample is. And then you can run a salmon directly on the overall FastQ samples, and that will save you a lot of computational resources. Okay. And then, yeah, so, with Callisto or salmon, you get expression index, and then you can, yes, as we mentioned, you check batch effect, because, it could be that these, I have seen data like this. They did tumor normal pairs, and you look at the data, they're always clustered in two. It turns out half of the samples were processed in the summer, and the remaining samples were processed in the fall. And that's clearly a batch, not really biological. So you might want to remove those and make sure that the remaining data really cluster based on the tumor normal in this case, before you try to see whether there is difference between the relapse samples and the non-relapse samples. Yeah, so one thing you might want to check is the differential expression between tumor and normal, because they already give you some difference, because individuals are still quite different after all. And so you could consider running the PCA based on the differential expression, because with the differential expression, you actually get the score from the DEC on all the genes, even if they are not differentially expressed, right? So you can get the robust readout for the differential expression and use that to run the machine learning part. You can also just try, if I use relapse or non-relapse samples to do differential expression, do I see some differences? You might already see difference already from this. And so based on that, you might want to run a cluster based on the differential gene, and also check whether those, also you can cluster based on all the genes that are variable, most of the variable genes between the samples. Then you run cluster on the samples, and you can check whether on the sample cluster, you can see whether the relapse status is focused on one cluster or the other. And then if based on the differential genes, you can run gene ontology or gene set enrichment, and you can see whether there are some other public cohort, and these differential genes are indeed associated with worse survival, because some of the molecular signature DB indeed will tell you this is a signature for previous studies of relapse in some other cancers, and that will be giving you more confidence that you are getting something interesting. And finally, you can run classification. This can be run directly on the tumor samples and to look at the difference between relapse and non-relapse. Or you can run this on the differential genes, which genes are differentially expressed, and you just collect all of those genes that are different between tumor and normal, and then run them machine learning. You can try all the different machine learning approaches, evaluate their performance with your cross-validation, and then use this to, and then you can examine the features, especially genes that are having high weight, and see whether they agree with your differential gene expression analysis. And then you can predict the new samples. And so in the kind of the exam type of setting, you know, you want to maybe say you want to do star mapping, map reads to the genome, run RCQC to get the quality control, get the expression index, PCA to visualize potential batch effect in the samples and remove outliers. Then you run DESeq or Lima to get the differential expression. This can be between tumor normal or it can be between relapse or non-relapse. And then you can run David on the differential genes based on tumor normal and also based on relapse versus non-relapse. So to run GSEA, and then you can, so in the meantime, you might want to run some heat map or PCA to evaluate this. And finally, you can run some different machine learning approaches, evaluate their performance using, you know, CARET or sklearn. I'm actually debating about this because this is R-based and sklearn is a Python-based. We might actually ask some students to try the Python-based in homework 3. We're still finalizing homework 3. And you can use this to pick the best method to use to predict the 5 samples. Okay, I see some questions from students. How would you actually remove samples that are bad? You just eliminate that from your analysis. So there are definitely cases when, say, you start from 200 pair of samples, and at the end you decide that I can only use 180 pairs, and the remaining 20 pairs will be eliminated from your study. And this, for example, could be determined by RCQC. Remember the MAD-10 score. And also, if some sample is such a crazy outlier, totally different from others, I would think that you would see it directly from your RCQC. But if by magic, if it works perfectly, but it's just, it passed QC, but it's such an outlier, just supposedly, maybe you have a sample swap. Your neighboring lab had some unrelated samples, you will see that it as an outlier. I would suggest in those cases, remove that sample completely from your downstream analysis. And sample here would be like one library that has been sequenced, not individual reads. Yes, in this case, sample would be one tumor or one normal, or it's just one RNA-seq data, either a paired end FASTQ in 2 FASTQ files, or if it's a single end, it's just the one FASTQ file with all the reads for that sample. So k-means is, you have to decide kind of your k. I think most of the time, you can run these machine learning approaches or some to determine k, but most of the time people just pick a k and they run k-means and see the difference. For PCA, I would say, I think most of the time people run PCA on the samples, whereas k-means, I think actually most of the time you can run this on genes. You want to see what are the major classes of gene behavior that happens between the groups. And especially in the time course, it gives you a very good idea. This is a general trend of a group of gene. This is a general trend of another group of gene. And k-means actually are used a lot more. They have very different purposes. PCA just project data onto 2 dimensions, but k-means usually you have kind of you use a heat map to visualize this, right? So it's a very different, yes, they are both clustering, but they are kind of quite different as well. Okay. So I also need to stop the recording.